This Week in Virology, the podcast about viruses, the kind that make you sick. From MicrobeTV, this is TWiV. This Week in Virology, episode 1269, recorded on November 7th, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hi, everybody. Actually, right now, so far, it's only you, Vincent. Hi, Vincent. What do we got here? It's sunny, and I looked at the thermometer, and it's 87, which is a little hotter than it's been recently, though it's pleasant enough. And my wife just told me that so far, it is the hottest November on record in Texas. I don't know if Austin specifically or Texas in general, but hey, this is where we're headed, right?
It's climate change.
Climate change.
Here in Chelsea, New York, it's 15 C and cloudy. It's been windy today. Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to see you all. Yes, it has also been very windy here. I am glad to not have blown away. And it is 58 Fahrenheit, 14 C here, and also pretty cloudy.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It's 54 Fahrenheit, 12 C, and also cloudy and windy here. Been like this for days.
Can you believe it? A quarter of November's already gone.
Wow. Well, it's less impressive to say the first week of November.
Yeah, tempus fugit, right?
Yes.
Yeah.
Oh, boy. Well, if you want to spend time with us, well, we like that, and we hope you enjoy it. And if you really enjoy it, we'd love your support, because like everything else, it costs money to make podcasts, and we would love your support to do that. If you want to hear real science, none of the misinformation, disinformation, lies. If you want right from the papers, we get the papers and we tell you what they say or don't say. If you like that, support us. We'd love it. Go to microbe.tv/contribute. This is the giving season, right? November, December. So please help us out. We don't do ads, as you know. And we are a nonprofit entity. So in the U.S., you can deduct your donation. Although I haven't been able to deduct anything for years. Used to be once, maybe you'll remember those days when you could make an itemized deduction, but then they changed the standard deduction so high that it never mattered anymore.
Yeah.
So no matter what I deduct, I can't.
Well, not only that, but they eliminated some of the major deductions, like your mortgage and other.
I think you could also deduct credit card interest.
Yeah.
And all kinds of interest. Yeah, they took it all away, and they handed it to the wealthy people.
Yep. Who didn't need it. And who don't spend it.
It's ridiculous.
I still go through the deduction thing every year anyway, because it forces me once a year to look at what I spent and add the whole thing up.
I don't like to do that. I don't want to look at what I spent. I want to be
blissfully ignorant?
Just blissfully ignorant. That's the phrase I was looking for. Microbe.tv/contribute. Maybe you are in a tax bracket where you can get a deduction above the standard deduction. And you know, if you donate X amount of dollars to MicrobeTV, it's not 100% deductible, right? Because there's always a fraction depending on where you are with your whatever tax return.
Yeah, but if you donated like a huge amount to endow the entire enterprise in perpetuity, you could probably get a pretty good write-off.
Yes.
True.
Yeah, people do that all the time. Wealthy people do that kind of thing all the time.
Yeah,
businesses benefit from that.
I mean, we would that would be great if someone could endow. I don't know how much it would take to endow MicrobeTV so that it could go on forever. You know, because all of us are not going to be here forever.
Well, what does MicrobeTV cost you? It costs you X amount of dollars. Okay,
yes.
If that's you can figure that an endowment will spin off 4% a year in spending money. Okay, so what?
Yeah.
25, 30 times whatever it costs you, that'll be the endowment.
Oh, 25 to 30 times.
Fair enough. That's Finance 101 coming from a dude who, doesn't know a whole hell of a lot.
Maybe there are people out there who know better. So yeah.
Send us mail. If you got a formula that's better than what I just said,
if our
send us
costs
mail
are
and money.
X,
Yeah.
Yes.
right? If our costs are X, how much do we need?
Yeah.
I know for professorships, right? We know how much you have to endow to pay a certain amount of a professorship's chair, but that's gone up over the years too. Anyway, I think like $2 million nowadays you can endow a chair, but it doesn't pay the whole salary. If it
My chair
spins
was
off,
a lot cheaper than that.
yeah, right. Okay, microbe.tv/contribute. Don't forget Immune 100 live at the incubator December 22nd, right here at the incubator, 6 p.m. It's a fundraising event, so that you need to buy tickets, but they're a donation, and you can tax deduct those as well. Plus, it's going to be a small group here, about 20, 25 people. It'll be fun. 2 guests, all of the Immune 100 hosts will be here. Should be fun. A couple of other things. Rich, what have you got to tell us?
So, right before I came in here to fire up the Austin TWiV studio, I read a breaking story in the New York Times that James Watson, co-discoverer of the structure of DNA, has died at the age of 97. So that is breaking news. And the article, actually, in the New York Times is pretty extensive. I think it's, and there will be more obituaries, I'm sure, coming out. He really is probably one of the most, if not the most recognizable name in science.
He's pretty high up there, yeah.
Yeah. In 1953, at the age of 25, as a postdoc at the MRC in Cambridge, along with Francis Crick, they figured out with very little data, none of their own. Okay.
Important note there.
Yep. Some X-ray crystallographic data that they had from Rosalind Jones in Maurice Wilkins' laboratory.
Rosalind Franklin.
Rosalind Franklin from Maurice Wilkins' laboratory. And also the important observation, Chargaff's law, that if you ever extracted nucleotides from any living material, the amount of guanine always equaled the amount of cytosine, and the amount of adenine always equaled the amount of thymidine. Those two central observations, and a lot of thinking and model building, which was novel at the time as well, led Watson and Crick to deduce correctly the structure of DNA, which was absolutely groundbreaking, not only for sort of its intrinsic value, but it immediately suggested how DNA could replicate, how the code could be copied, and also gave very significant insights into what was then the growing understanding of how the genetic code worked. So this was a paradigm shift as big as anything that we've witnessed in science for, centuries, decades at least. Quite a discovery. 25 years old. He went on to run his own laboratory for quite a period of time at Harvard University.
What did you say, 25 years old? What? No.
25. At the age of 25.
Oh, at his age of 25.
His age
okay.
of 25.
Okay.
He went on to have his own laboratory at Harvard for a number of years, and then took over the directorship of the Cold Spring Harbor Laboratory, and was mentor to quite a few reputable individuals, contributors to science over a period of time, not the least of which was my mentor, Joan Stites, and several others. His life was not without controversy, but I'm not going to detail that here, because the man has died and he contributed quite a bit to science. If you want to read about the controversy, which is worth reading about, I'll refer you to the science article. So rest in peace, Jim Watson, and thank you for your contributions.
Trying to think what I was doing when I was 25.
25.
50.
I was a graduate student.
I was in your lab, Vincent.
I was also a graduate student.
Is that right? Oh, you were in my lab, yes.
I was in your lab when I was 25, not when you were 25. No,
come on. So 20. What's 53 plus 25? 78. Right? So 1978, I was 25. I was a graduate student. Yeah.
I was. It was 1973 for me, and I was in Jones' lab. Probably 2/3 the way, 3/4 the way through my PhD work.
I was in kindergarten.
When
Brianne wasn't born yet, right?
Yeah, I was gonna say, in which year are we talking about?
Yeah, 73.
Brianne, you were haploid?
I was haploid.
Yeah.
Yeah. What were you doing when you were 25?
I was a graduate student working on CD8 T cell memory in 2005.
So we all were doing similarly things.
So young.
Similar things at the age of 25. I guess the career of a scientist is almost collinear, with some exceptions, of course. But you do things in a certain order. That's quite interesting. All right. So next we have the Russian translation of TWiV 1024. That's the Chumakoviridae.
Awesome.
So Olga, who is from Russia and was trained there, wrote, and she said, I think this is a great example of U.S. Soviet cooperation. So this was when Sabin went to Russia, Albert Sabin went to Russia to test his oral poliovirus vaccines. And he did a big clinical trial there. And Chumakov, in that interview, is his son. And we talked a lot about that. And she said, this is a great example of how, even two countries that don't get along politically can still collaborate scientifically. And it's lost on today's Russians. So I want them to read it. So she translated it to English. And she got Chumakov to correct the things that it was just to correct it. So it's now published on Medium. So I'll put a link to that. It's really great. Thank you, Olga, for doing that.
Nice.
Yeah. What is it? How do you say thank you in Russian? I don't even know.
Здравствуйте? Or
What?
Здравствуйте?
Something is
that thank you, or is that good
spasiba.
morning?
I have
My
a
wife
clue.
would know. She speaks a little of it.
Thank you, spasibo.
Spasibo. That's it.
Spasibo. Spasibo, Olga and Poka. Okay, that means goodbye, I think. We have a few people who speak Russian on the live stream, and they have taught me a few words, which I promptly forget because I don't use them. That's the way it goes.
That happens with language.
Okay, Alan, you're next.
Yeah. So a couple of items I pulled up out of my feeds. Canada has a functioning government, which is apparently still working, and they just passed their federal budget for the coming year. That budget has measures to aim to lure foreign researchers. They didn't specifically call out the U.S., but it's very clearly tailored to lure researchers in the U.S. who would like to go to a place that respects science. They did have to cut a little bit from their science budget, but not nearly as much as people had feared, given their financial situation. And they are funding it and inviting folks from other places to come in and contribute to Canadian science, eh?
Okay.
So,
It's cool.
something to think about there.
Yeah, a functioning government is something novel.
Something to think about.
Yeah.
Yeah, I was so confused when you said this word, like federal budget agreement.
Yeah, some of their federal government did something. Are they allowed? Yeah, some countries they are.
I'm resisting the urge.
Yes.
I don't know why, but I am.
Yeah, that's all right.
That's because we're a science podcast.
Yes.
Well, the only thing I want to say is food is science. and you need to give people food. And I don't understand the desire not to feed people. This makes no sense to me. But nothing else makes sense that this
No.
administration does. Nothing, okay? And feeding people is the most ridiculous. Actually, going to court to say why you shouldn't feed people doesn't make any sense. Okay, what's next, Alan?
Okay.
Mini rant.
More
That's nice.
more good news. Bird flu. So it's migration season. If you've heard geese flying over your house, you know this. The wild birds are all flying south down the major flyways and especially down the Midwest going, down to South America. They are carrying with them H5N1 flu and spreading it to poultry along the way. There are more cases popping up, but we don't exactly know how many, and this isn't being monitored very closely at all because the CDC is, the people who still work there are mostly, not allowed to do their jobs. And so bird flu is now surging in various parts of the middle of the country. Past 30 days, it struck 66 poultry flocks, killing 3.5 million turkeys, chickens, and ducks. And we are largely blind to the full scope of this because we're not really paying attention.
So Daniel said the other day that I think it's one of the Dakotas has the most turkey farms in the U.S.
I think that's right.
But I think it's Washington, D.C.
Yes. Yeah.
Most turkeys are in Washington, D.C.
Very good.
I saw this great video of a cat eating from a bowl, and this chicken comes up and tries to get some of the food, and the cat, bats it. The chicken backs off, and then the cat starts, and the chicken goes right back. Doesn't give up. It's really cool.
Both parts of that sound entirely correct.
Yes.
I think interactions among animals are very interesting. There are lots of videos on Instagram. You don't know if they're all real, but the interactions amongst. So I saw one of a person feeding a carrot through an opening in a zoo, and two, I don't know what they were, capybaras were fighting over it, and so one would get it and the other would grab it back, and then the one would get it, and then finally one won. And the other one was like looking at it, and then it turned around and looked at the hole, and the person put another one. It's like, where's mine?
They know the drill.
Yeah. I loved it. It's great. It may not be real, but it's entertaining. Okay. What else is up, Alan?
Got one more flu story. So part of monitoring influenza, as we've talked about on TWiV, is trying to figure out what strains to put in the next flu vaccine. Typically this is coordinated globally by the World Health Organization, which sends samples around, and individual health officials in different countries largely send them to the CDC because they have the capability to do the typing on the strains and figure out what's spreading where and help coordinate this. See above comments about the current situation at the CDC, and in addition, the United States pulled funding from the WHO. So those two things together mean that we are not only blind to bird flu, we are also largely blind to which strains of human influenza are spreading and are likely to end up with a bad match in the next flu vaccine.
That's unfortunate because I think this year we got our strains from we got the strain composition from WHO, but we were lucky because we're not doing anything now.
Yeah, I mean they will still come out with a strain composition, and the vaccine companies will make it, but we won't have as good a set of data as we normally would. And this article explains what normally happens and what's happening now.
Explain to me why we cut the WHO budget again. I don't understand.
Somebody didn't like it.
Because
it's science.
We don't like people outside of the United States.
Yeah, and because it's science, as Rich said. That's the best I can come up with. It doesn't make sense to me either.
If there was a way that people could see the transcript of comments in my head about a number of the stories we have spoken about today, they would be very, very appalled by some of the how annoyed how mean I've been with some.
I'll tell you what, Brianne, you should start a podcast where you do that, and it'll be great. It'll be billions of people listening. Where Brianne rants. Anyway, it's really unfortunate.
I'm willing
So,
to come on as a guest if it.
Perfect. Perfect.
Well, Dixon and I were going to do that. He said, "Why don't you and I do a click and clack, 15 minutes a week?" I said, "Okay," but then he got sick, unfortunately. But it would have been fun. I saw an ad where, I don't know, what one of these AI companies, either ChatGPT or Gemini or whatever, I don't know, they were saying, the future is science and research, and we have the tools to help you. It was a whole freaking ad about research and science, right? So this is not the what's the word I'm looking for? The climate has nothing to do with climate change. The climate is that science is good, but there are a minority of people who are trashing it. It's really unfortunate. That's what I meant to say. And I also saw another ad by Pfizer talking about how important it is to get vaccinated as you get old. Right?
I think that there's some, I think Pfizer ad that I've seen that's really just about like celebrating science, and it's such a great ad. And
these are ads that people pay a lot of money to put on TV, you know, or whatever, streaming, whatever. And so obviously there's an audience for it. So it's very puzzling to me that you want to trash science, because one guy somewhere thinks that it's a good idea. The guy who wrote the 2025 plan, right?
Yeah.
He thinks it's a good idea. And so we have to go by what one frickin' person thinks? That's not how it works. It's called a democracy. We did have an election, though.
Yep.
And people spoke. Okay, we have a snippet. We're going to continue. We're now on an arc with human papillomaviruses. Two episodes constitutes an arc, I guess. And so after
it's
last
at least
week's,
a line segment at this point. It may turn into an arc.
I don't know what's the requirement for an arc, but I guess extended.
You know, we can make our own definition.
That's right.
And we're running the show here.
Somebody said to me in the live stream the other night, it's better if you teach and don't talk about politics. And I said, you know what? Don't tell me what to do. You can correct me. You can say, oh, you're wrong. And that happens a lot. And that's fine. I understand. But if you tell me what to talk about, that's really bad.
No.
Okay? Because it's my program.
sometimes I try not to talk about politics in class, but
I wasn't mean. But then he apologized. He said, I was just trying to make some joke. I said, okay, that's fine.
Usually
But
about day 3 of the semester, I fail.
Yeah.
I don't like when people tell me in the middle of a program what I should talk about, right?
Well, you know, I would rather not talk about politics. The problem is that it now intrudes on too many things.
Well, it was on science particularly. And many people then retorted to that person, no, you have to talk about it as it impacts science. And it does, and always has, and it always will. So, you know, when politicians make stupid decisions, but the decision to withdraw funding from WHO, to withdraw funding from USAID, what's the other one? Gavi, PEPFAR. I mean, all this doesn't make any sense.
No.
Makes zero sense if you look at it from a scientific public health perspective. It's an ideological decision by a few people who have a certain view of the world. And you know what happens when a few people have a view of the world and they get in power? This is really a problem. So
I'm all of a sudden, this is peripherally relevant, but I'm all of a sudden reminded of the two quotes, one from Peter Daszak, I think, and the other, I forget where, we've gone over before. One is that public health, by definition, is a mashup of science and politics.
Yes.
And the other is that when you mix science and politics, you get politics.
Yes.
You don't get a nice blend. That's right.
No.
Okay, Alan, you want to do there's a paper. The paper is a PNAS paper from 1983.
Yes.
When I was 30 years old. I just started at Columbia the previous September. Human papillomavirus type 6 and 11 DNA sequences in genital and laryngeal papillomas and in some cervical cancers.
And the first author is Lutz Gissmann, and the senior author is Harald Zerhausen. This team, it's only 6 authors, actually. They're from Universität Freiburg and the Universitätsfrauenkliniks at Marburg and Düsseldorf. in Germany.
So last time we talked about a very cool meta-analysis where the conclusion is, even if you're older and even if you already have, at least in women, if you already have HPV infection, it's worthwhile to get vaccinated. There's a 75% chance that the infection will be cleared. And even if you have a lesion, it helps to get vaccinated, particularly before the surgery, because it helps your prognosis. So I thought we would do the origin of HPV and its association with cancers. And this is just one paper of many that Harold Zurhausen co-authored. Of course, he won the Nobel Prize in 2008 for his discovery of that human papillomaviruses cause cervical cancers. And so if you go to the Nobel site, you have a lovely history. By the way, it's the same year that two people got the prize for identifying HIV-1. And so this starts in many years ago, people thought that cancers of the anogenital tract, and particularly cervical cancer, were caused by an infectious agent, sexually transmitted. And a lot of people thought it was herpes simplex type 2. So Zurhausen was working on this, and he never found herpes simplex type 2 DNA in cervical cancer cells. And he used a technique called in situ hybridization, where he would take sections of tissue, say, and treat them appropriately. And then you can add a nucleic acid that is labeled. And then if there are any complementary nucleic acids in the section of the tissue, the probe, as we would call it, would hybridize to it. And then if you exposed it to film, because back then it was radioactive label, you could see the radioactive dots over the cells. So he never saw any simplex 2, even though the technique works beautifully, and you can certainly use it to see simplex type 2 DNA in other cells. So he said, why don't we look at papillomaviruses? There was some evidence that, they cause warts, of course, papillomaviruses of all different species, but the human papillomaviruses cause warts in humans. And he said there are genital warts, anogenital warts, and maybe and even oral warts and laryngeal warts and respiratory warts. He said maybe they're caused by HPV. So he started to look at that, and he didn't have a lot of. I mean, he had in situ hybridization, but what are you going to use? You couldn't grow the virus, so what are you going to use to do your in situ hybridization? So he had to extract RNA from cells, actually, and use that. But and then eventually he purified some viral DNA from a plantar wart, right? There's a kind of wart you would have on your hands or feet. And he showed that there was a signal in cells from these warts. And other people started to work on it as well, and they started to get some evidence that in cervical cancers there were DNAs as well. And so this paper, by the way, he eventually provided strong evidence that HPVs were involved. He discovered multiple different genotypes of HPV, as we'll see in this paper. And then he and others showed that early region proteins, in particular E6 and E7, is that E6 and E7? Yes. Mm-hmm. Get integrated into cells and cause the cells to divide and eventually become cancers. And even experiments were done putting those genes into cells and showing that they transform cells. So eventually that was the proof that these viruses cause not just cervical cancers, but oral cancers and anogenital cancers and cervical cancers. There's a very nice page at the NCI which discusses. So we have anal cancers, we have cervical cancers, we have oropharyngeal cancers, also called throat cancers, tissues of the oropharynx, and that's different from oral cavity cancer, which forms in the mouth. They both have head and neck cancers. Then we have penile, vaginal, and vulvar cancers. The vast majority of these are caused by HPVs. So Zurhausen got the Nobel Prize for that. And then subsequently, of course, vaccines were developed, which really proves it because the vaccines prevent cancer.
Yeah.
Right. So if you needed more proof, because there's no animal model, and it's very hard to grow the virus in culture. So anyway, that's the background. And so this paper is, as I said, 1983. And they say, he says in the introduction, maybe genital cancer, he calls it, or has an infectious etiology. And we're trying to see if papillomaviruses are involved.
This paper is really kind of one of like a major milestone in virology, because it's not exactly the start of this idea, but it's the real beginning of figuring out the scope of it.
So previously,
I think I read this either in graduate school or in college.
Mm-hmm.
The paper looked really familiar. I said, yeah, I've definitely read this before.
So they had previously characterized a papillomavirus in genital warts. They called it HPV-6. So condylomata acuminata, okay, those are genital warts. And he found HPV-6, which I think 6 and 11 cause almost all of the genital warts that do not proceed to cancer mostly. They're mostly not proceeding to cancer. But they're such a big cause, so it's not considered a high-risk papillomavirus. But they're in the vaccine. They're in Gardasil 9 because there are just so many of them, and it's debilitating to have genital or oral warts, especially in the larynx. They can impede you from breathing, right? Mm-hmm. So why not have a vaccine that prevents it? So that's why they're in there. But he had previously found 6 in these genital warts, and had looked at other malignant tumors and didn't find it there. But more recently, he found they cloned DNA from a laryngeal papilloma. So now cloning is a thing. Molecular cloning is recombinant DNA is possible. It wasn't possible when he first started. And so that DNA, he says, shared about 25% of its sequence with HPV-6. So they called it HPV-11. So that's the genesis of HPV-11. And so this paper now tests the idea that the laryngeal papillomavirus actually have a genital origin. And they also look at tumors for the presence of these DNAs. So it's very straightforward. And the materials and methods are 3 short paragraphs.
It's 2 inches of column space. And
right?
Yeah.
They tell you how to extract DNA from cells. Biopsy materials were examined, histologically stored at -20 or 70 until further processing. Extraction of cellular DNA was done as described, reference 6. So this is a feature of older papers. You did a reference trail. You go to 6, and maybe it would tell you, but maybe it would refer you to another paper.
Right.
And you'd sit in the library for many hours.
Tracing back to figure out how to do this
because
damn
there was no
technique.
online stuff.
Yes.
Right? You had to get the actual books, and at the end, your hands would be dusty and everything.
You also, in a lot of these old papers, get a bunch of information that's now in the materials and methods in the figure legend.
Yes.
That's true. Labeling of DNA. I love this because this is something that people don't do anymore. The HPV-11 DNA was subcloned in PBR32 at the BAM-H1 site, which is what I used to do all the time with other things.
PBR322 was like the first cloning vector. Something you said early on, Vincent, was that in passing, was that you can't grow HPV, which is still essentially true, except for very sophisticated and complex culture systems. So we talked last time about how the virus infects the basal cells of the epithelium, and then the program of gene expression with the virus is altered as it migrates through the epithelium and encounters differentiation signals in the epithelium. So you can't, in one cell type, simply reproduce the whole virus replication system. It needs all of these differentiation signals. So that's why you can't just slop it onto cells and have it grow. So I don't know, but I imagine that this is one of the first papers where the virology is largely, if not totally, dependent on cloning techniques, which were relatively new at the time. And I also, they talk about all these restriction enzymes, and I'm sitting here thinking to myself, they don't say we got our BAM-H1 from New England Biolabs. Somebody down the hall was purifying this stuff.
Yes.
That's right.
Yeah.
Oh, nowadays you have to have a table of everything you got and from where, right?
And while I'm at it, the other bit you mentioned was that there was evidence that the genital cancers had an infectious etiology, specifically that they were sexually transmitted. And I forgot that. I had forgotten that, but that's absolutely key in nailing this down. This looks like a sexually transmitted disease.
Yeah, in fact, there's a quote by Zurhausen in the Nobel thing. In 1976, he said, "The condyloma, the genital wart agent, has been entirely neglected thus far in all epidemiological and serological studies relating not only to cervical and penile, but also vulvar and perianal carcinoma. This is unusual in view of the localization of genital warts, their mode of venereal transmission, the number of reports on malignant transition, and the presence of an agent belonging to a well-characterized group of oncogenic DNA viruses, HPVs," right? So that's very interesting. By the way, I did interview Zurhausen a number of years ago. I'll put a link to that in the show notes. We're just standing up somewhere face to face.
Yeah, I was just looking at
talking
that.
to each other. It's not too long, and you should check it out. It's fun. It's a long time ago. Of course, he died not too long ago. And the last method I want to tell you, because this is important, is it's called blot hybridization. And so David Baltimore would say, blot is jargon. You can't use that word. It is jargon. Do you know? I mean, if you go to a person on the street, do you know what a blot is in science? No. A blot should be, Southern blot hybridization is the whole name of the technique, but it's still jargon. You take DNA. So in this case, they're taking either cellular or viral DNA. They're going to cut it with a restriction enzyme, and then they run the products on an agarose gel, which separate by size, right? And then you take the agarose gel, which is relatively thick, right? It's, half a centimeter thick. And then you have this elaborate procedure in the beginning anyway. You'd put it on a stack, a sponge, and then the sponge would have a wick going into buffer, and then you'd put paper towels on top. Oh, I forgot the nitrocellulose you'd put on top of the gel. And then you'd have this fluid wicking up, sucked up by the stack of paper towels, and then you put a brick or something on top. And the DNA would transfer from the gel to the thin sheet of nitrocellulose. If you walk through labs, you'd see these homemade Rube Goldberg things, right? Where you've got like a
Pyrex baking dish, man.
Pyrex baking dish,
Yeah.
and it's got buffer in it, and then there's a sponge, and then you've got your gel, and then there's filter paper so that the fluid can wick up, and then stacks and stacks of paper towels. You know, those brown multifold paper towels people used to.
And if you're really cool, in addition to the weights on the top, you put your favorite little icon. Like,
That's
Yes.
I would probably I've got a little statue of Anthony Fauci here. I would probably put that on top.
And you'd let this wick, overnight typically. And then subsequently they had electrical methods of doing this, right? Electrotransfer.
And it's called a Southern blot because the guy who invented it was named Ed Southern.
Right.
And then, just for kicks, as long as we're at it, and talking history, years down the road, they figured you could transfer RNA. They, being Jim Allwine, figured you could transfer RNA and probe it the same way. And being a humorous individual, he decided as a pun, he would call it a northern blot. That's where all that comes from.
And then the immunologists got in on it and started probing protein gels, totally unrelated technique, really, protein gels with antibodies. And they decided, those nucleic acid folks are doing it. We will too. So that's a Western blot.
And there was never an Eastern.
There's never an Eastern blot.
There was never an Eastern blot.
There's still an opening there.
Yes.
Okay, so this is a prominently used technique in this paper. I mean, only a couple of figures. So they want to be able to differentiate HPV 6 and 11 in cells and tumor material. So they say we have 3 ways that we can do this. We can cut the DNA with BAM-H1, this restriction enzyme, and it cuts HPV 6 and 11 just once. Remember, the viral DNAs are circular double-stranded DNA. So if you cut it once, it becomes linear, and then it will migrate in the agarose gel according to size. And so they say, if you probe with, if your probe is HPV 6, and you run both 6 and 11 on the gel, the 6 will be brighter. It was hybridized more strongly than the 11. You can see that in the first figure.
So, yeah, I want to make a comment here about typing human papillomaviruses, because ordinarily we talk about serotypes. And if you, for serotypes, what it means is that you've got a virus and you've got antiserum, and you look to see whether antiserum A reacts with viruses A and B, or the other way around. But with human papillomaviruses, we already said you can't grow the virus. And in fact, they induce a really crappy antibody response. So you don't have virus, you don't have antibody. And so what they did was to do hybridizations of whatever DNA they could get under various conditions, and make some sort of estimate of how much nucleic acid homology there was. This is before sequencing. We can't actually sequence.
I was just going to say, why didn't you just PCR it up and sequence it?
Yeah.
No, the answer
No
is the paper
PCR,
is
no
1983.
sequencing. You have to do things like melting under different salt concentrations, or what they do here is they wash blots with different temperatures and different stringency conditions, and you can make an estimate of how much sequence homology there might be between one virus and another. And they kind of put their heads together and came up with rules as to how much difference in sequence homology would be required to say that one virus is a different type relative to another virus. Right. And the thing that blows my mind is that way down the road, it turns out that now, since you can create vaccines and artificially induce sera, but more importantly, create vaccines and immunize people, that all of that typing turns out to be true. If you have a vaccine against what you're calling HPV 16, that gives you immunity to HPV 16, but none of the others. So it all worked out. Amazing.
Yeah, in fact, in the introduction, they say this. So they found 6, and then they found another which shared about 25% of its sequences, and they said that's the rule of making a new genotype. So they called it 11, 25%.
When I saw that sentence in the introduction, my first thought was, I wonder how long it took them to come up with 25%. Like how much work that took compared to, the afternoon it would take.
Well, and I think it also, these were rules that evolved over time as the techniques evolved. And I've seen some, I used to know more about this, but I've seen some where ultimately they focused more on the major capsid protein, L1, which is appropriate because that determines the, in quotes, serotype, et cetera. But they got it right, basically, all the way along.
Yeah. So it's the same with poliovirus, right? The original definition was serologically based, but now they sequence VP1 and they put it into type 1, 2, or 3 based on that. That's it.
And so the reason, for those who might be confused, they went from 6 to 11 because 7 through 10 had already been found by others?
I don't know. That's a good question.
I don't know.
I was wondering about that, yeah. So I think, let's see, it says in the Nobel thing what the numbers were here. Where is it? Initially they got 1 to 3. 4 was described a year later. This is all about not cross-hybridizing as well in southern blots, as we have just said. So that gives us 1 through 4. And then here, Zerhausen gets 6 and 11. I don't know what happened in between. Then we go to 16 and 18. So I'm not sure.
In a quick Google search, it does say that Zerhausen is actually the one who described some of the ones in between.
Yeah. Okay.
And I just looked it up. There are now identified over 200 types of HPV.
Yeah, it's amazing.
And about 40 of these can infect the genital area. And still HPV 16 are the major cancer causers. They're the highest risk.
Yeah, in fact, I found a nice chart in the Nobel lecture showing the contribution to cervical cancer of the different types. And 16 it accounts for 53%, and 18, 17. So that's 70% of all cervical cancers. And then 45, 31, et cetera, accounts for the rest up to 99.7%.
No, I think this has mostly to do with the activity of the main oncogenes, E6 and E7, right?
Yeah, I was going to ask about what you thought. You think it's all about
I
E6?
think that's the major deal, but I can't, I can't be absolutely certain.
I mean, it may also have to do with the person and the genetics of the
I wouldn't
person.
be at all surprised
Right.
if this is one of those things where if you looked it up, you got a variety of answers,
Yeah.
which always says to me that we really don't know.
We haven't quite worked that out yet.
By the way, as I had predicted, Daniel picked up last week's paper and talked about it on the clinical update.
Excellent.
And he said, Rich, you did a great job describing the progression of the lesions.
Oh, good. Thank you. I appreciate that because I've been doing it for years, but I'm still sort of skating on thin ice out there.
You used to teach it, right?
Yeah, this was one of my main deals because of Kath, among other
Yeah.
things, and because it's interesting,
Yeah, it's very interesting. Oh, so Silverstein, who
important.
was my next door neighbor, used to work on these as well. And I got so tired of hearing about coelocytes or whatever they're
Oh,
called.
that's another interesting thing. Okay, so in a pap smear, if you do a pap smear, you scrape some basically cells off the surface of what actually Daniel, check me on this, but the diagnosis is of a cervical dysplasia
Right.
is done, I believe, by by spraying, if I'm not mistaken, a little bit of dilute acetic acid on the surface of the cervix, and for reasons that I don't understand, what is essentially flat wart tissue shows up as a different color, I think sort of whitish, and you say, this is not right. And you don't have to do that before you do a pap smear. When you do a pap smear, you just take a swab and swab some stuff off the surface of the cervix and spread it out on a microscope slide and stain it and look at it. You're basically doing cytology. And the clinical marker is what is called coelocytes. I've never been able to pronounce it correctly, which are characterized by having fairly condensed nuclei with a halo around them. You can see space between the nucleus and the nuclear membrane, and they're very characteristic. And dude, that's a cytopathic effect. Okay? The virus causes a cytopathic effect, as many viruses do. Lots of, in old-time virology, you would identify what virus it is by what sort of cytopathic effect it caused. And I always thought of this as, this is a pathology of a papilloma. But yeah, it's the virus cytopathic effect that's showing up that's showing you this pathology.
Just so people know from some quick searching, there seems to be quite a bit of evidence that the difference between high and low risk HPV is very much in the biology of E6 and E7.
Okay.
I found searching that comes up pretty quickly
Okay.
in terms of exactly what's going on with the E6 and E7 of those particular strains.
Those are the more oncogenic E6s and E7s.
Okay, back to the paper. So we're doing southern blots. We're getting stronger hybridization when you go across genotypes, and also you can wash at different temperatures and see that you've got different genotypes. And then the third one is, which is the best in my opinion, you cleave with another restriction enzyme, PST1. It cuts it multiple times. Sure, you get different pattern fragments for the two genomes for 6 and 11. I mean, that's just easy, right?
Yeah.
And so you could take viral DNA and this is the beauty of a southern blot, you could take total cellular DNA, right? Billions of bases, and cut the whole gamish with a PST1 and run it out on a gel. And if you stain it, you get a big smear of DNA from big to small. When you probe it on a southern blot, you get bands for HPV. You can pick up those bands in the whole background of cellular DNA, which is the amazing thing. Now we're all jaded and we say, yeah, so what? But it's really cool. I mean, imagine being able to look at individual genes that way. That's why Southern blot was so revolutionary.
Yep, it was revolutionary.
And so they used this third approach to screen 63 typical condylomata acuminata warts of anogenital origin. And 86% of them had either 6 or 11. And they found 6 3 times more frequently than 11. So this is now why 6 and 11 are in the vaccine, because so many warts are caused by it. You just want to prevent them while you're preventing cancers as well. The genome, so you can get a rough idea of how much DNA is in each cell by how many cell equivalents you load on and so forth. 0.1 genome up to 100 copies per cell of papillomavirus DNA. One sample they had DNA that they could find under reduced hybridization stringency, so they figured that's a different type. That's how they, as Rich said, that's how they figured out the different types. And in 5 specimens, they didn't find any sequences, but they're probably there because they're so distant that they didn't hybridize at all. Okay, so then they have 5 flat or atypical condylomata of the cervix and vagina. They all had HPV-11 in between 10 to 100 copies per cell. And 11, by the way, that was originally isolated from a laryngeal papilloma, okay? That's not the genital area, folks, okay?
No.
It's far away. It's up here, laryngeal.
Other
larynx,
end. Yep.
Okay. They found it in low concentrations and 5 of 12 more cases of laryngeal papillomatosis, so confirming that it's present in those. They have 1 case of tracheal papillomatosis, so further down into the respiratory tract. So again, the idea that other kinds of tumors are also associated with these viruses. We can't say caused yet, right, because we don't have enough evidence. So we say associated, because this is basically an observational study. 24 malignant genital carcinomas, they were tested, and they did this PST digestion, and they found 1 pattern in 1 cervical carcinoma. So that's the data.
Yep.
Those are the data.
And the paper is open access, as well it should be, being 40 years old.
So basically, you have a new type of HPV from a patient that had recurrent laryngeal papillomatosis. So this is the thing. They can recur. So you can remove it surgically. They'll come back, unless now we know you get vaccinated. You can probably prevent them from coming back. So that's 11. That's the new one from a laryngeal papillomatosis, and it's
These
in this
go
paper.
up to 11. Yeah.
They find this in 7 of 14 laryngeal and tracheal biopsy specimens.
I forget whether you mentioned this, but another sort of connection for them was that one school of thought, at least, was that laryngeal papillomas were acquired at birth from women who had papilloma infections of the genital tract.
That's right.
The laryngeal papillomas are no joke, because there can be many warts, and they recur, and they have to be lased off repeatedly, and it's a nasty disease.
Yes. So again, associated with 6 and 11, so that's why they're in the vaccine now, because it prevents that nastiness.
Awesome.
But they also find them in genital warts, as you saw in this paper as well. So it's the same virus doing it at different sites. Now, the other thing they found is that 3 of the 24 malignant cervical tumors, they have 1 invasive and 1 carcinoma in situ. They were positive for HPV 11. So they're saying, we don't know what this means, but we need to study it more. So they're in the warts. There's no question that this virus is in the warts, but they find a few tumors that are positive, and they say it's hard to assess the significance of these data. Additional studies are required to elucidate the role. And as I said earlier, eventually studies were done.
Those got done.
It was
many
proven,
by Harold Zerhausen.
and we have a vaccine, and you should. Yeah, it's a wonderful vaccine, and the people who disparage it are really out of line, completely out of line, because it prevents cancer. It saves lives. By the way, this is what I wanted to tell you. I didn't tell you. there are an estimated 700,000 cancers globally every year caused by HPV, and 350,000 women die from cervical cancer alone.
Preventable.
Totally preventable.
Now preventable.
I mean, people who are, come on, stop with the nonsense. This vaccine does nothing but save lives. I wish people would just get that.
Yeah. The immunology is also so nice on this vaccine.
Yeah.
And sometime we'll do a HPV vaccine paper.
So this is good science here. You already mentioned this. At present, it is hard to assess the significance of these data, and additional studies are required, blah, 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 as opposed to this is absolutely the best science ever done, and this is the answer to everything, and we did it.
How much of that is driven by reviewers now? Like, how much
Some.
of that, would sort of a humility-like statement like this get eaten up by a reviewer at this point, and that's why they've gone away?
So it's a good question. That's a very good point. I think in the old days, people did it themselves. They policed themselves. But things are tough now, and you have to push yourself, right? And so I think reviewers are playing a greater role. Right?
Yeah, on more than one occasion, though, I've been out of the business for a while. I've seen outrageous claims in papers that had the effect of lauding the authors. And I said, dude, this is inappropriate.
Yeah, I
Take
agree.
this
No,
out.
totally.
Yeah.
One thing used to drive me crazy. You read in a paper, this is the first time this has been
Yeah.
shown. And I was like, what the hell? You're publishing it. Of course it's the first
Of course,
time.
the first time, yes.
You don't have to say that. And there was such arguments about this. In fact, one journal would not let you say,
Yeah.
this is the first time. And mostly we don't see that, but it's just ringing your own bell. Okay, we have a Journal of Virology paper, Influenza virus infection and aerosol shedding kinetics in a controlled human infection model.
And the
Alan.
authors are two co-first authors, Nishit Shetty and Meredith Shepard, and the senior author is Seema Lakdawala. And I should add there is a correction published for this subsequently. They forgot two authors who were in the middle of the list.
Ooh.
Yes, Andrew Catchpole and Alex Mann, who now I'm mentioning because they were forgotten before, and their affiliation for those two is HVivo in London, UK. The affiliations for the other authors are on page 16. However, and I want to call this out, ASM has done something very good here. They provide a link to page 16, so you can just zip down to it.
That's good.
Yes.
Good job.
I also took note of the fact that they mentioned that the editor is Stacy Schultz-Cherry.
Yes.
I appreciate
So
that.
these folks are from Virginia Tech in Blacksburg, University of Kansas in Lawrence, Emory University School of Medicine in Atlanta, Duke University in Durham, Emory University regular campus in Decatur, University of Michigan Ann Arbor, and University of Pittsburgh School of Medicine, and HVivo in London.
So thank you, ASM and JV.
Yes, good
for
job.
putting a link. Other journals, you should consider doing that. It's not so hard.
Either that or just put the author affiliations up at the top. It's not that difficult.
Yeah. So I was really excited to read this paper, partially because some related things have been coming up in some of my classes, and students have been asking things, and I often have to talk about, okay, how would we actually know that information? How would you do the experiment to know that information about transmission of a particular virus or shedding or things like that? And so I think that any person who doesn't actually think about these experiments for this very often would look at information like this and be like, oh, obviously that's a thing we would know. Like, obviously this is sort of super basic textbook thing that everybody knows. But in fact, experimentally, some of these questions are pretty hard to deal with. And this paper is great because it actually does some of the important things experimentally, but pointing out that it was done in 2024. We are just now really having the ability technologically to answer these questions that I think most lay people would imagine, you didn't know that? What do you mean? So I really liked reading this paper and sort of seeing this kind of basic key information actually be elucidated.
Yeah, I mean, because the thing they're investigating it really is, how does influenza virus spread?
Mhm.
And yeah, I'm totally with Brianne. Like, yeah, you're right. We didn't actually know quite how that. We've got some ideas. We've got some evidence that points in directions, and this is the way you do the experiment, is with they had to come up with an abbreviation for controlled human infection model, which they call a CHIM.
Yeah, I shaking your head, Vincent.
Well,
I had trouble with that too.
so the reason they did that is they don't want to call it a challenge because that has a bad name, a bad rep, right? A human challenge, because you're challenging these people with viruses. So a human challenge would be short, but they don't want to call it a challenge because that's politically not
They
good.
should call
So
it controlled human infection model procedure.
It's like calling the MRI machine magnetic
resonance imaging, because you can't use the word nuclear.
Exactly.
Yes.
Right.
Yes.
See, whereas you were thinking chimp, I was thinking about Mary Poppins and, Chim Chimney.
Oh, wow.
Yes,
That's
good.
better. I like that.
Anyway, so here's the numbers on flu. It's 290,000 to 646,000 deaths per year globally caused by influenza virus. Okay? So that's, like Lennon said, one death is a tragedy, 1,000 is a statistic. So you look at those and go, okay, it's not a lot, but somebody in there is related to you, or is your kid or your parent. You got to be careful with numbers like that. Anyway, it's a lot. And so we don't know a lot about transmission, kinetics of shedding, period of contagion, and the modes of spread.
Yes.
Those are some of the questions that this paper is trying to address by these controlled human infection studies.
They note that the epidemics are burdensome.
Yes.
Yes, they are burdensome. Now, there have been others done before, but they often do not measure infectious virus. They don't examine the quantity in the aerosols and so forth. So they mentioned 2 human challenge studies. Oh, there's the challenge word.
Yes.
Wow. They found infectious virus in respiratory samples, but they didn't look at the dynamics of spreading. So that's what they're doing here in this study. So this was done I'm going to do a little bit of the materials and methods, which are substantially longer than the previous paper.
Yes.
This goes many, many pages. This was done at Emory University Hospital from July to September of 2022. And how do they infect these people? This is important. A dose of 5.5 log 10 TCID 50s, tissue culture infectious dose 50% in 0.5 ml, was given to each nostril using an atomization device, a intranasal mucosal atomization device. I guess they're spraying it in, right?
Yeah, as long as they're not, shooting 0.5 ml into your nose, I wouldn't enjoy that.
Yes.
No,
and they say the patients were in a supine position when this was done. So.
Yes.
Yeah.
So but this is the first point, is that that's not how you typically acquire influenza virus. And they admit that in the discussion, and they say we have to work on that. On day 0, they are admitted to the hospital, and then they're there for 8 days for the duration of this study. Okay, that's one method I wanted to do. The other is respiratory aerosol sampling. I mean, there's swabs and so forth. That was all standard. But they collect aerosol samples on the day of admission and study days 2 to 7. Basically, they have a galvanized aluminum funnel with a 20-centimeter opening connected by tubing to a machine that's going to capture particles and condense them and make them into a liquid.
This is a little like the Gesundheit.
Yes,
I know.
It's a
I
little
was
like the
thinking about the
Gesundheit.
Gesundheit. Yes.
I was
So whether
figuring that I was figuring that they were going to have the Gesundheit machine in this.
More or less similar. So basically, they put this on your face and you breathe for 10 minutes. It's called tidal breathing. You just sit there calmly breathing. You should meditate probably. And then They get rid of the medium and replace it. And then you speak for 10 minutes. Here on TWiV, we have no problem speaking for 2 hours.
Well, I was curious about that. Like, what do they say? Do they just talk to they're like, please talk to this funnel for 10 minutes?
Or they give them something to read?
Yeah, did they give them something to read? Did they like recite something? Did they basically have a rant about politics? This is going to be the first episode of my ranting podcast.
You know, the plosives give you more of an explosion,
right? Exactly.
Right? So maybe they have plosive-rich text.
Yes.
With a lot of P's. Besides the P, what else is a plosive letter? T probably is a little plosive. Yeah. T. Okay. All right. So that's the sampling. And then they do...
B.
B's another one. They do digital droplet PCR. I don't think we've talked about this before, but it's basically PCR. It's RT-PCR, right? So that it's viral RNA, influenza viral RNA. They can make a DNA copy. And then you have a volume which is put into a machine which disperses it into many, many, many tiny, tiny droplets. And each droplet is assayed for the presence of DNA. And each droplet gets either yes or no, right? A 0 or 1. That's why it's digital.
So you're doing basically 10,000 PCR reactions,
yes,
each in a very small volume of droplet.
Right, it's all in the droplet, and the machine makes the droplets, right? So that's different. That's something we haven't done, but it's considered to be, more accurate than other methods. And I think everything else. Okay, so we have 51 people originally screened. They were 18 to 49 years old. They were screened for susceptibility to the challenge strain, which is an A. Perth 2009 H3N2 virus. 13 who had hemagglutination inhibition titers less than or equal to 40 were taken, and the rest were sent home if they had higher titers. And they were screened for other criteria. And eventually 8 people were enrolled and put in their own rooms a day before getting virus.
So how did it go? Oh, it sucked. I got ruled out from getting flu and being in the hospital for 8 days. Darn it.
Yeah, but probably they were paid a good amount of money.
Yes, there's compensation for this.
And they collect daily nasopharyngeal swabs, and they test for virus by PCR. And so now we see some of the results. 6 of the 8 participants were PCR positive on at least day 1, and they had symptoms consistent with influenza infection, which we'll talk about in a moment. So they have a 75% attack rate.
Yes. And just for people who were concerned about this, they also mentioned above that any participants who at the end of the 8 days were still positive, or AKA hadn't had 2 consecutive negative tests, were given oseltamivir. So they treated these individuals before releasing them from the hospital.
Yeah.
Right. So I'm going to do something I usually don't do, which is refer to figures, because this is open access. And the figure 2 is interesting. It's viral shedding in nasal and oral cavities. Now, let me just say something first of all. Why don't you have a time 0? You had to just take another sample and assay it. Don't tell me you needed to pay for more people, because you didn't. And in this one, panel A, on day 2, it's already up there. I don't know if that's replication or if it's the inoculum. That's why you need a day 0. Okay?
Yeah. Good point.
And some of the others, so panel C, which is infectious virus and saliva, on day 2, it's baseline. So, okay, you could get away with not having a time 0 because it's baseline. And then everything after that, it goes up, it's replication. But, nasopharyngeal CT value, panel A, infectious virus, the ones that day 2 are not baseline, you just don't know.
So can I ask a silly methodological question that I've only just thought of as you're saying this? And so you might be like, no, that's a really dumb question. If you did that swab at day 0, would you worry about that basically removing some of the dose? Or would you worry that then you're not actually giving consistent inoculum to all of your patients because you're taking some of it back out from some?
I would do that swab an hour or 2 after I gave the inoculum.
Yeah, you could do it. you have a window before replication's happening, right?
This
It would
reminds
actually
me.
be- It would be day 1, by the way, just so we're...
Yeah.
Yeah, it would be day 1 inoculation. Yeah.
Reminds me of doing a time course of infection in a plaque assay, right? You do the adsorption and then you wash the monolayers and get all
Yes.
the other stuff off.
Yeah.
And then you take your time 0 thing, and that's whatever virus that you can see around in the adsorption. And then you take other time points and hope that you see an eclipse, that stuff uncoats and goes away and then comes back. That would be the awesome experiment. I don't even know if that'll happen in a challenge model.
It's a good question. I mean, in the old days, we always used to do time zeros. Okay?
Yeah.
I don't know. Now the current thing is no more. I don't understand the logic behind this. It makes 0 sense. It makes 0 sense.
0 cents, yes.
But also, they have their time 0 is the day they come into the hospital.
Yeah.
Right. And then day 1 is inoculation. I'd do a day 0 just to see.
Yeah.
Might actually be a good idea.
It wouldn't cost that much more. And I'd say maybe on the way in, they pick up something in their nose. Who knows? Who knows? I'm making good jokes today. Anyway, so you see nasopharyngeal. They divide these people into high shedders, low shedders, and no shedders. Okay? They call them non-shedders, but I call them no shed. I think that's more interesting.
And
they're looking at
shedders. Yeah.
nasopharyngeal swab. They look at PCR, CT values, and infectious virus, and then saliva, infectious virus, and then nasal lavage, infectious virus. And so you can see, if you're looking at nasopharyngeal swab, infectious virus, at day 2 you have something, but I don't know if it's inoculum or replication. At day 3 you really have some replication in the high and in the low shedders. Okay, so day 2, after you put this in people's noses, when they start shedding, and then they shed for 5, 6 days, depending on whether you're a high or a low shedder. So that's your window for flu, right? But we're going to talk about symptoms in a minute. All right.
So they have, I'm getting picky here, but they have, I'm thinking about a similar figure from a challenge study that I used to use in teaching flu.
Yep.
They, in the infectious virus, in the nasopharyngeal swab, it's measured in PFU per mil. So that must be per mil of the swab having been rinsed out in saline or something like that. I want to know what the titer is in PFU per mil of snot. Okay? And I don't think you can get that from this.
No.
Because I want to know, I used to do this calculation with this study I used before. I want to know how much virus is in a sneeze. Right? But the numbers are pretty big here. I got 10 to the 5, 10 to the 6, even up to almost 10 to the 7th in one of these points. There's a lot of virus coming out of the nose here.
Yeah.
Yeah, the nose is where it's happening. Yep. So 4 participants had multiple days of PCR positivity with 1 sample of CT value less than 25. So the cycle threshold, right? The more cycles you need to see a signal on the PCR, the less RNA there is initially. So a high CT, there's not a lot of RNA, but a low CT. So 25 is okay. I mean, let's see, what is the scale here? They're going from 45, which is nothing basically, all the way up to 0. And so these are like anywhere from 45 to 15. So 15 is a respectable gangbuster CT, I would say.
Yeah, you only have to amplify it 15 times to
15
see it,
times, yes.
instead of having to amplify it 45 times before you saw it.
So these people who had multiple days of PCR positivity, including at least 1 sample with CT less than 25, are called the high shedders. And then 2 people are low shedders. They have a different criteria. And then 2 people remain PCR negative for the duration. So these 2, it's very interesting. These 2 people got virus in their nose. There's no question about that. They're not on the graph.
Nope.
Right? So there is where a day 0 would be great. Maybe you didn't stick it in their nose. Maybe you turned your head and it went somewhere else. I realize that's not likely.
No.
But if you had day 1 showing the inoculum, I'd feel a lot better about that.
Maybe they sneezed.
I think they should report it if, in fact,
Yeah.
2 people sneezed, and so they probably sneezed everything out. Anyway, that's what you have to have a day zero. I don't want to hear any arguments against a day zero.
Okay, we're not giving you any
Not from you
arguments.
guys. You guys can argue all you want. But I know the authors are going to argue, TWiV, this is why we don't need a day zero. No, I'm not going to listen.
Well, we should listen to the arguments, but I doubt we're going to buy them.
They will likely need to be extraordinarily convincing before
Yes,
you will.
there needs to be absolutely compelling.
Now,
And
wouldn't you like these non-shedders to see a day zero, a peak of virus, and then boom, it goes down? That would be fabulous. And how long does it take to go down? Well, you don't see anything we don't know because they didn't put them on the graph because they said it was negative. So I guess by day 2, it was negative. Anyway, that's...
by the day after inoculation, it was negative. So what you should see is day 1, something... Rich's ideas sound wait an hour after you've actually put the virus in there. So you've had a turnover of the mucus. Then sample. There should be something in there. And then,
So we used to,
comes down.
And I'm sure you guys did this. You take a plate of cells, you remove the medium, you add the virus inoculum for an hour or so. You add the medium, and then you take your time zero.
Yep.
So you've given the virus time to absorb. If you want, you could wash off the inoculum because by then everything's gotten in.
That's what we did. We washed
That's
off
your
the
time
inoculum.
zero.
Yep.
Right? See,
And there was always, and you always got a number.
Yep.
Right? And then
Sure,
later
absolutely.
on, the eclipse is so much fun because it's cool to see that number go away. The virus is uncoding. And now you're down to 0, and you know that everything you see after that is new stuff.
Okay, so here's a actionable piece of data. The peak of shedding, this is viral RNA and infectious virus, between 3 and 5 days after inoculation. Okay, now in the real world, you don't know when you're inoculated, right? But you're going to learn in a minute when we talk about symptoms. And there are differences in detection. Like one low shedder had infectious virus from nasopharyngeal swabs, but not from nasal lavage fluid. Shedding of virus in saliva was sporadic, so saliva is probably not a big vector of transmission. But this is interesting. Both the low shedders had infectious virus in their saliva, but not elsewhere. So remember, saliva is generally low in everybody, but in these 2 low shedders, they had virus in their saliva, but not elsewhere. So sampling a single site, they say, may not capture the full spectrum of virus shedding during infection. So, you get a nasal swab. That's how we do it clinically, right? And even if you may not be positive, but you may still be infected. And if you check somewhere else, you might find it. But it's, that could explain some of the discordance that we see.
Yeah.
Even with other viruses, I would guess, too. Then, we often use RNA in wastewater to look at viruses and gauge their extent in the community. So they decided to look at, as they say, viral RNA in excrements, stool and urine samples. So the high shedders had detectable RNA in stool on days 6 and 7. There's a genome copies per milligram between 380 and 9,000. And these days correspond to the detection of infectious virus in the nasopharyngeal swabs, but not in the other sites collected. But they didn't get kinetics because not everybody pooped every day. That's the reality.
Yeah.
They don't say it that way, but I'm saying it that way.
That pretty much captures it, Vince.
Stuff happens, but not reliably. Yes.
No viral RNA in the urine above the limit of detection.
Urine's probably a pretty hostile environment for flu.
Yeah.
There's a control for you. Take some flu and mix it with the urine and then titer it, right? See if the urine kills the flu.
They got some RNA in fecal. So they had 6 fecal samples, and 2 of them were positive towards the end of the quarantine period. And these people who had positive stool had also higher virus titers in the saliva, and they say probably that's how it gets in the intestine, right? You're swallowing, and that saliva with virus in it is getting down there. But they say we need to have a better study of excretion of viral RNA. This was not good enough. I don't know how you're going to do that. You got to ask people how often they poop, and then you take the ones that poop every day. I don't know.
I mean,
Yeah,
this,
that's
I
an-
think part of
an-
it is just having a much larger study.
Yeah.
But
Maybe.
Bigger
So
group.
regularity is an inclusion criterion?
Yes.
So, would any of you guys volunteer for this kind of study?
I don't think I would do this.
No, I wouldn't do it.
No.
No. I'm too old.
I mean, they'd probably look at my birth date and say nope.
Yeah, you're too old. You're at risk for more severe infection. That's the problem. Now, Brienne is not, but you still don't want to do it.
no, I don't like having the flu.
Yeah.
And spending a week sitting around a hospital room is not going to be thrilling.
Yeah. Would you do TWiV from your hospital room?
Of course.
Oh, yeah.
Yeah, if you.
Okay, here's a good one. Disease symptoms.
Yeah.
Okay.
This is good.
So they score symptoms daily. And the most common symptoms, and you should remember this next time you get sick, runny nose, nasal congestion were the big ones, and headache. That's it. Runny nose, nasal congestion, and headache. Now, there could be other things. You could have scratchy throat, you could have fever, whatever. But runny nose, nasal congestion, and no coughing. It's interesting, isn't it?
Well, I'm trying to remember. They did sort of a funny thing that I remember reading in the methods, where they were afraid that people were going to sort of have subjective ideas
Yes.
about runny nose, congestion, scratchy throat, sore throat, chills, felt cold. And so they actually kind of did sort of averages. So they asked runny nose and congestion, and they averaged what you said for runny nose and congestion and sort of made that its own thing. And so part of me kind of wonders about, where does coughing fall into some of these combined symptoms?
Yeah, because they're surveying people for their symptoms, which is how you would have to do this. And people will read the question, and one person will say, "Do I have a runny nose?" Well, I don't know. Do I have congestion? No, I don't have congestion.
Yeah.
But
Yeah, it's hard when people self-report stuff.
yeah,
It's
So-
too bad you can't.
They didn't report body temperature on any of this?
They didn't.
No.
That's kind of
I'm a
strange.
little surprised about that.
yeah,
They must have been taking their vitals every day.
sure.
Sure. Now, let's see. Let's check out the methods here briefly.
If this was an animal study, you'd report weight loss.
Weights, yes.
Yeah. No, if you look at figure 1, which is a timeline, there's no biometrics. None whatsoever, unless I'm missing them. Let's see.
That is an odd omission.
Clinical symptoms. Yeah, it's this stuff,
It's all
scratchy
using
throat.
this diary.
Yeah. So here, among 32 symptoms, there's 32 potential symptoms. They picked 11. Runny nose, coughing, headache, body ache, tiredness, chills, feeling cold, congestion, sore throat, scratchy throat, and sneezing. So there is chills, which could be indicative of fever, right?
Mm-hmm.
But they don't
But I
actually
mean, a
measure.
thermometer would be more reliable.
A thermometer might be reliable, yeah.
I mean, I would hook them up to a BP and a temperature.
Yeah.
monitor.
I can't believe the data aren't available.
In the hospital, that's as soon as you get in the
Yeah.
room, they
Yeah,
wire you up, right?
Yeah, and then they come in the middle of the night every night and wake you up.
Right.
And
but
Yep. They do. All right, where were we here? Okay,
So we're on-
symptoms. So the thing about symptoms that's interesting is that the symptoms occur. So this PCR positivity,
they only partly correlate with viral loads.
right? But they occur a day prior. The PCR positivity is a day before your
Right.
symptoms. So you're potentially shedding virus a day before you feel symptoms. So the day you get symptoms, okay, you've been infected for a couple of days, maybe 1 or
Right.
2 days.
And you maybe even infected other people today.
Yeah,
Yeah.
so
right.
you could sort of time the onset of your infection by when you get your symptoms, and then you configure another 7 days or so before you're really not shedding anymore. maybe 5 days. It really depends, right? You just
So
don't
that's
know.
some of the most practical thing that comes out of this is
Yeah.
that I often wonder when I'm at the peak of symptoms now of a cold. I don't, does this, I don't know, different diseases may behave differently. This is flu, but I often wonder when I'm at the peak of symptoms, am I past the peak of virus shedding or not? This suggests that there's a pretty good correlation
Yeah, the peak
between the peak of virus
symptoms
shedding and the peak of symptoms.
correlate with the peak load, viral load. Yeah.
And it's important, as you point out, that when you first start feeling symptoms, you've probably been certainly infected a couple of days before and possibly shedding virus.
Yeah.
Yeah. So they say the highest symptom scores were on and peak nasopharyngeal virus lows were observed on study day 4, which is 3 days post-inoculation. So there you go. And then after that, it goes down. And the high shedders had more and persistent clinical symptoms compared to low and non-shedders.
But not, I mean, the numbers are small, but
Yes.
there are some outliers. I mean, number 039 had significant symptoms, but was a low shedder.
Yes, it's very
And
interesting.
007, sorry, that's the guy's number. Only male in the study it was a low shedder and had low symptoms.
Yeah, I
Which
actually
correlates.
find the most fascinating part of this the non-shedders.
Yes.
Because
Yeah.
the non-shedders did still report some symptoms.
Some symptoms.
And so that, trying to figure out what was going on with the non-shedders, was there a little bit of infection or was there, really not seeing a lot of infection going on? This might be a part of it, but also they knew they were in a study where they got something squirted up their nose. And so trying to figure out what's going on with those non-shedders, especially, 10 seem to mostly have sore throat.
Yeah.
So what's up with that? And it does make it seem like, perhaps some infection was occurring in these individuals given these symptoms.
Well, 10 reported sore throat on day 1, the day of inoculation. And then it kind of goes down after that, which actually made me wonder if this is somebody who had maybe a little rhinovirus infection when they showed up.
Now, what would be cool is if we had like their interferon levels at these times.
Yeah.
And
Yes.
if you had interferon in your nasal cavity when you came in, did you become a non-shedder?
Yeah, that would be easy enough
Yeah.
to do, right?
Those data are probably available too. They
Or if
did
you
blood
had
draws.
or if you had ISG like transcription in that location, did your ISG levels correlate with whether you ended up being high, low, or non-shedder?
As a matter of fact, they've got serum samples from screening at minus 60 days from admission, day 0, and then a couple of times during the infection and at discharge and afterwards. So those data ought to be available.
I'd like mucosal
Yes.
Yeah, absolutely.
interferon, right? Yeah,
Mhm.
that would be cool. Okay, now shedding. So these respiratory explosions that we described, they look for viral RNA, and they find the average amount of viral RNA that is expelled during speaking is greater than during breathing. Isn't that not surprising, right? You do expel some during breathing, but much more during speaking.
This was a big deal during the pandemic.
Yes.
Because we talked about
Oh yeah,
singing
Yes.
right.
as
Yes.
being one of the really hazardous things to do.
Yep. Yes, sir. Singing is even worse than speaking, probably.
Now to have these people sing.
It's sing into the funnel.
Yeah.
Well, isn't that what
Well,
they did in those COVID gazoon type
I-
studies?
some-
Is that
yeah,
I'm
I
pretty
think
sure they
yeah,
did have them
yeah.
sing like "Twinkle Twinkle Little Star" into the funnel.
But if you have ever seen a play and you happen to be close, you know that when they speak
Yeah.
loudly, they're expelling a lot of
Yeah.
Yes,
stuff.
play that I saw recently, the way the lighting was seemed
Yes,
to
the-
really particularly highlight just the
Yeah, they're really
stream.
projecting, right?
Yeah.
'Cause- yeah. Okay. Anyway, viral RNA was found in breathing emissions for 2 high shedders and speaking emissions for 3 people. 2 high shedders and 1 low shedder. Only 1 high shedder expelled virus during speaking that could be detected across multiple days. And that person also had higher infectious virus in their saliva. No infectious virus was detected in any aerosol samples. They say probably there's not a lot there, and also it gets inactivated in the collection process. And then they do they do. So that's an interesting point. No, they didn't detect any infectious virus in any of these respiratory exposures. That's probably a methodological issue because I'm sure there is some there. That's how it's transmitted, right? So there has to be some infectious virus there. All right. So then they look to see if the amount of viral RNA correlates with the number of total respiratory particles. So this would be droplets expelled. And they basically find no correlation. So I'm not even going to talk about that. They sampled the environment. They sampled 4 sites: the floor under the bed, the mirror. The mirror is a funny one. The tray and the bed buttons.
And the logic of the mirror is when you're brushing your teeth, you're likely splattering some of your oral contents onto the mirror.
So it's the bathroom.
In my house, it's not likely. It's happening.
It's definitely happening. Yes, I wasn't going to point. Yeah, I when I clean the bathroom mirror, that's
exactly.
Well, when you use an electric toothbrush,
that would make it even worse. Yes,
it does it.
But even a manual toothbrush is going to.
We'll do it also.
Splatter.
All right, so they sampled those from days 2 to 7. Look for viral RNA by PCR. They only found RNA in 2 high shedder rooms. The tray of 1 with a CT value of 33 and 28, so there's not a lot of RNA there. So they got days 4, 5, and 6. And then the bedside control buttons for another person on days 4 and 5. Again, CTs of 37 and 35. Very little infectious virus there, I would guess, because those CTs are really high, right? Everything else was negative for viral RNA. And they did not look for infectious virus.
Do you think this is about how long this virus persists on the surface? Maybe if you had just looked at the surface after someone directly sneezed on it, you'd find something. But if you actually sort of wait for the amount of time, and think about how long that is going to persist on the surface, you're really not seeing much.
I think that's
over time.
a legitimate question. Yeah.
Yes, I think so. So you could do an experiment. You could have someone put their fingers in their nose, right, and then touch 6 buttons. And then every 3 hours, you take a button and swab it because you can't use the same button.
Oh, see, I was just going to have them lick the button.
Jeez.
Try getting that past the IBC. Okay. Anyway, I would say from this that environmental transmission is probably not a big deal.
We didn't think it was a big root, and this adds to the evidence.
On the other hand, if you are a skeptic, you say, but TWiV, they didn't find any virus in aerosols either, so it's a kind of a limitation, right? But we do know that influenza virus is transmitted by respiratory droplets. I would say from this, it's probably not fomites. How about seroconversion?
And just backing up a little bit to the aerosol issue, the sampling method that they used really seemed to emphasize aerosol rather than droplet.
Yeah.
Because you're breathing or talking into this funnel that's connected to a tube that goes down into the sample.
Yeah, it's true.
Rather than, talking to a sample plate or something that's going to take droplets. So it seems like maybe you ought to look at droplet transmission too.
Yeah, and so basically if the droplet fell and hit the funnel before it made it to the tube, then it wouldn't be counted in that study.
Yeah, right. Okay, then seroconversion. A 4-fold rise in hemagglutination inhibition antibody titer is what's considered the threshold for saying you've seroconverted. So all 4 people with high shedding had a 4-fold rise in HA antibodies specific for the virus, while people with no detectable virus loads had no evidence of seroconversion, and neither the 2 with low viral loads had a 4-fold rise, even though they were shedding virus. So guess it wasn't enough. And the neutralization assay, which they also did on these samples, is basically the same. And then the last thing they did is they wanted to see if there was any original antigenic sin going on here. So basically, when you get infected with an influenza virus, it can induce the production of antibodies that were against the very first influenza virus that you ever saw, your first flu.
Yes.
So they looked for so these people are being challenged with an H3N2, right, from 2009. So they looked to see if they made antibodies against an H1N1 from 2009, the pandemic virus. Nada. Nobody had any antibodies against H1N1, all against H3N2. So there was no sinning in those 8 hospital rooms. Hey, there you go. I think there are a lot of interesting takeaways.
Absolutely. And going back to Brienne's, although we've, been chuckling all through this because it's, there's elements of this that are amusing. Sorry. All right. But it's an important study. And going back to Brienne's initial comments, it emphasizes that getting these kind of data and understanding fundamentals of infection and transmission of a disease like this in humans is not easy.
Yeah, and as much as we've pointed out, little gripes about this, like please take a day 1 or day 0 sample. Overall, I think this is a hell of a lot of work, and I'm really glad that they did it.
Yeah.
And I am looking forward to other types of studies like this. I mean, they even say we need to do more like this with this type of thorough analysis on larger patient populations because 8 people, there's a strict limit to what you can conclude.
There's so much useful information that's coming out of this.
So this makes me think of a study that I think we've talked about before, Vincent, not as a paper on TWiV, but I think in talking about it, you and I may have been at the same Gordon conference where some guy gave a talk on transmission of, I don't know, it was a rhinovirus or something like that, talking about human subjects where, just like the ferret experiments, he compared subjects where you had an infected person and a sentinel person that were either separated in different cages. I mean, they were rooms that had screens in them, so they couldn't touch each other, or being in the same room. All right, and it was a fascinating study that concluded that for that particular disease, which I think was some sort of rhinovirus infection, that it required that people be able to actually touch each other to get efficient transmission.
That's right.
From
For rhinovirus, yes. Actually, the classic rhinovirus transmission, you have people playing cards.
Ah, okay.
You're sitting at a table, and one of them is infected, and then you watch the infection spread just by the cards.
That immediately reminds me of you and I going to interview Fred Murphy at the beginning of the pandemic, and we went to his. He was living in a retirement residence, and walking through the lobby to get to his room, and there's a bunch of people sitting around playing cards. You and I are looking at each other going, whoa.
There's a high risk
I don't know
area.
if we ought to be doing this.
That's right. So this is the thing, like, the one thing that I don't get is the inoculum. Like, they're putting basically 1 million TCID50s into the noses, right? But in real life, you're going to get a whole range.
Yeah.
I want to know what is the threshold, right? And it's going to be different for different people also, right?
Yeah.
Everybody's going to be slightly different.
Yeah.
And so maybe you could do an experiment where you put people in a room and you have different rooms, and in each one you give them a different amount, and you see who transmits to others based on the original inoculum. It's very complicated.
Yes.
I think either study, the study that you talk about or the study that Rich mentions, both have the sort of challenge of statistical power.
Yes, sure.
Because as we look here, they put a decent sized dose directly into the nose of these patients, and only 6 of 8 were shedding. And so if you actually start thinking about, well, okay, so only some fraction of those who I infect are going to shed, potentially only some fraction of those they come in contact with are going to acquire the virus
Right.
after shedding, and on and on and on. At some point, you have to have a pretty big number of individuals to look at any of these questions about thresholds or things like that. And so I know that in other settings, you tend to use a relatively large dose because you want to get your 8 people infected. If you did this study and 0 of 8 got infected, then that would kind of be sad for all the work you've put in.
So here's a question for you. Let's say you use different doses. You have 1 million, and then I would do 1 million, 5 million, and 10 million. Okay, 3 sets. Would you expect the kinetics to be any different?
We've
Because
talked
there are
about
people
this
during
before.
COVID. People during
Yeah.
COVID said, "Yeah, the more virus you get a different
not only
disease."
the kinetics, but the severity of the symptoms.
Yes, exactly.
And I don't know the answer to that.
Because we know in this case the symptoms kind of correlated with how much virus you're shedding,
Right.
But it doesn't say anything about the inoculum.
That's not right.
But that would be an interesting question because you remember during COVID, people were saying if you get a higher inoculum, you're going to get more severe disease. And
I don't buy that.
Don't know where the evidence for that is.
And it just,
Well, I guess the question is, would you get more severe disease or would you just get earlier disease?
Well, you can't increase the reproduction cycle of the virus, right?
Right.
It's 12 hours or whatever, but you put much more virus in, it's not going to accelerate it.
right.
So
no,
I
I think you either get it or you don't.
I think
And the size of the inoculum affects your odds of the infection taking hold.
probably.
But I really don't expect the size of the inoculum to make a big difference in the severity
So
of
I
the
would
disease
say-
because that's not what determines the severity of the disease.
You would get the same kinetics, but maybe the peaks would be higher with a greater inoculum. And maybe everybody would be infected with 5
Maybe
million,
everybody would be
Right.
infected.
right? But that would be interesting. I mean, so I think playing with the inoculum is important. I also think playing with natural transmission. I don't know if you can do that in a flu study. I don't know if that's allowed, right? To let people, like they do for the rhinovirus transmission, let people transmit it to each other. That would be very interesting.
Yeah.
Because here, and also monitor the shedding like they did here to say, okay, so this person sheds this much and they didn't transmit, and this person did. But of course, transmission is also the recipient, so it's really complicated.
Yeah, so you'd sort of have to have for each potential shedder, you'd have to have multiple recipients probably.
Basically, we have to have an indoor soccer game and just infect
Yeah.
half of the stadium and see what happens to the other half, right?
And all have them live in the hospital for 8 days
Yes.
and have all of this monitoring.
I mean, more people, the better. But even with 8, it's very challenging. 8 hospital rooms is a lot of money.
Yeah.
yeah.
Anyway, it's cool. Good study. All right, let's do some picks of the week. Brianne, what do you have for us?
So I think this is a repick, but a repick from a very long time ago. So this is a board game, in my mind, because I actually had my students play it today, called Pandemic. It is a cooperative game where players play together to try to stop 4 different epidemics that are happening simultaneously around the world, each player having different abilities and roles, and you have to collaborate to try to beat the infections, and it's quite a bit of fun. We had a really good time playing it today, and I highly recommend it for anybody who is looking for something fun to do.
Yeah.
Can we play this on TWiV?
Maybe
Or do you have to be in the same room?
you.
It kind of,
It
I
works
played
best if
this.
you're
Yeah.
in the same room.
Yeah,
Yeah,
I'm not
I've
quite
played
sure how we could do it not in the same
There
room.
are platforms for playing board games and card games
Online.
through a medium like this, but they're specialized for a particular type of game, and I'm not aware of a.
Yeah, I don't know if there's one for Pandemic.
It would
Yeah.
be fun, but
It
if
would.
we're
It would
ever
be fun.
if we're ever in the same room, we should play it.
Yeah.
We should.
Okay, Rich, what do you have for us?
I have a New York Times article that got, shunted into my TWiV picks folder some time ago. Actually, it's from a couple of years ago. Entitled, Whiskey Fungus Fed by Jack Daniels Encrusts a Tennessee Town. And it highlights what apparently is a thing that's been going on for centuries, which is that if you do a lot of distilling in some place, you are likely to be infested with a fungus, and I can't believe there's only one of these. There's probably more than one that that thrives on alcohol. And in this town with a Jack Daniel's distillery, this black sooty fungus is like everywhere, and the residents are complaining about it and having a big to-do, or were, with Jack Daniel's. And there's some pictures here that are really pretty impressive of how annoying this might be. And the distillery, they argue that in fact the fungus contributes to the quality of their product, and so they don't want to do anything about it, right? So I just thought it was an interesting, an amusing read, and I'm thinking all the time about the fact that humans are trying to destroy the planet, right? And anytime I see an example of some organism growing under unusual circumstances or in a hostile environment, it just warms my heart, because I know that no matter how bad we screw this up, DNA will continue to replicate, okay? My money's on DNA.
First
Humans
evolved,
are-
last extinct.
Yeah, humans are incidental to the whole
So,
thing.
Rich, I think people don't try and destroy the world. They want to try and make money, so they ignore
true.
environmental concerns just to make more money. And then this is the outcome.
They're not deliberately trying to screw up the world, but their priorities are misplaced.
Yep.
So
Agreed.
in reading this, I was fascinated. And just as Rich said, I was sort of like, is this one fungus or are there many? So of course I just Googled it
Okay.
to try to find out, because I noticed that the New York Times article does not mention a genus or species name of what fungus. Apparently it's one particular fungus
Oh, okay.
called Bodonia companiensis. And it has a preference for airborne alcohol, as Wikipedia says, otherwise known as the angel's share,
Yes.
the alcohol that evaporates off.
They do mention that in the article, the angels share.
Yes, the
Yeah,
angel's share.
as the whiskey ages in the barrels, the angel share evaporates off, and after you have aged it and then you bottle it from the barrels, the last dregs that have the kind of the soot of the charred barrel in them, that's the devil's cut.
Oh, okay. Wow.
I do love this quote here from the mycologist they contacted, James Scott in Toronto, who says, "The fungus is pretty destructive, and the only way to stop it is to turn off its alcohol supply." And I think we can all think of people who would fall into that same description.
That's great. Alan, what do you have for us?
So this was actually a competitor for my Halloween pick, but it's another. I'm linking to an article in Science in the news section, and they also have a link to the original paper. So some researchers studying bats in a hibernaculum, an urban hibernaculum, near the entrance of a cave in Germany. They were filming with infrared cameras because it's dark, bats flying around, and they discovered that there are rats living in this cave that snatch the bats out of the air and eat them.
Wow.
So if you've ever, been a little creeped out by rats, and then you reassure yourself by saying, yeah, but they're rodents, they're herbivores, they're not really that dangerous. Well, maybe either do or don't watch this video, because these rats are predators.
I'll say,
Yeah.
this is really something.
And I found this fascinating because, first of all, I thought of rats as herbivores, and, maybe they'll eat a bug here and there. It had never occurred to me that they would snatch a bat out of the air and eat it. And secondly, this has some significant implications for disease spread.
Yes, yeah, it does.
I mean, my first thought is rabies,
Yeah.
but I don't know if
cool.
if rats get rabies.
I think there are a number of things, though, that both bats and other small mammals can share.
Yes.
It's interesting.
Pretty cool.
My pick is an article in Nature. Well, not an article. It's a news feature by Michael Marshall. 7 basic science discoveries that changed the world. So this is an attempt to illustrate why the science cuts in the current U.S. government, the gutting of U.S. science, are ridiculous, because basic research, and the work they're canceling is a mix of, applied and basic research. And basic research, he writes, is easily mocked because it can seem impractical, but in fact, it's a major driver of economic growth. And they have a number of examples here that you can read about. The first one is the discovery of bacteria that live in hot springs. Like, why would anyone want to do that back in the 60s, Tom Brock? But you isolate a thermostable DNA polymerase, which leads to your ability to do PCR, which runs everything nowadays, right? So come on, don't question things you don't know about.
Yeah.
MRI is another one with origins in research into the physical properties of atomic nuclei, root vegetables and flat screen TVs, chemicals from carrot roots. CRISPR, of course, is a great one. But this is one I really like. Weight loss inspired by a lizard, Ozempic, right? Well, a Gila monster provided the basis for GLP-1. And who would say, oh, what are we studying Gila monsters for? What are we studying worms and fruit flies for?
Because the more we know about the universe, the better off we are.
So these are great. If you ever have to give people examples of why you should not cut basic research, you can just read this and remember some of them, because this is just disgusting what's going on.
This is a really useful article. I'm definitely going to be using this one with my students.
This is also an awesome picture of a Gila monster.
Yes.
It's cool.
It's very cool.
Yeah, the story there is very, very cool. All right, that's TWiV 1269. You can find the show notes at microbe.tv/TWiV. We'd love to get your questions, comments, picks of the week, TWiV at microbe.tv. If you enjoy these programs, we would love your support. Microbe.tv/contribute. Alan Dove is at alandove.com, turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
Brianne Barker is at BioProf Barker on Blue Sky at Drew University. Thank you, Brianne.
Thanks. I learned a lot.
Rich Conda is an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.